Hi, welcome back. So what I'd like to talk to you just very briefly now is another aspect of the analysis on your bacterial genomes that you'll conduct later on. I want to just give you a little bit of, background understanding of what's going to be going on when we try to annotate our genomes, and what I mean by that is how taking these contigs that we generated in our assemblies, which are really just strings, sometimes hundreds of thousands of nucleotides long, of G's, A's, T's, and C's, and how are we going to look at those and read them and understand what genes are there. So, inferring function from DNA sequence. Well, basically what we do, although we do this using computer algorithms, and so you're going to use a really wonderful tool called PROCA in your analysis, but basically the principles that are used in that algorithm are the basic cell biology processes, and it's understanding how does a cell read a DNA sequence and make proteins, for example. And by understanding that process and in reverse engineering it, we're able to write programs that can predict the coding function of DNA sequences very well. And so you'll see that when we do this. This is, of course, aided by the fact that there are some, like all things with informatics, there are rules, and although we love the variation in biological systems, in these cell biology and genetic systems, they're near universal. And so we know, for example, that we used to call it the universal genetic code, but it still remains nearly universal. So there's this single set of information that allows you to know how an RNA sequence is read to produce a protein sequence. And it's read, 3 nucleotides at a time, generally starting from an ATG codon and ending in a stop codon, in your cases for your bacterial genomes. And so it's that kind of thing. And the fact that we know, transcription is very much like replication in the sense that how these sequences are synthesized and how that relates to the production of proteins is something that we can model in a computer and understand. We also understand that these sequences evolved from common ancestors, and so that evolution and that process, while it results in changes to the sequence, and in some cases changes to the proteins sequences that are predicted, it allows us to make comparisons from one species to another. And so when we've demonstrated the function, doing actual experiments from one organism, for example, in a model organism like E. coli, when we find a protein sequence that we know and can demonstrate is homologous in our bacteria, then we can, right or wrong, and sometimes it's not as simple as that, we can apply that function to that sequence. And so these processes, as we talked about, we know, for example, when we have a gene or a genome, that either of those strands can be used as a template strand for transcription. And we know how transcription works. It's a very collinear process. It's a very, at some level, it's a fairly simple concept to know that, our DNA sequences are represented by a single strand and a FASTA file. By processing them, for example, in ways that replicate the processes of transcription and translation, we can make predictions about all of the possible protein sequences that can be generated. And then we can ask things like fairly straightforward questions about, so what could be a gene in a sequence? And so if you think back to your biology class, we know, for example, that particularly in bacteria, genes typically start, the protein coding part of a gene particularly starts with a single amino acid, and that's methionine, and we know what the codon is for methionine, and in fact there's only a single methionine codon, and then, so it's an AUG. So we could simply look at all of the places in the genome or in the transcriptome of both strands where there's an AUG, and then we can ask questions, computationally of course, like, so if I was the cell and I was translating that from beginning with that AUG, how far could I go reading 3 nucleotides at a time or 1 codon at a time before I run into the stop signal, one of the stop codons? And so, we can ask that very rapidly using DNA sequences and programs that are running on computers, and rapidly blast through a bacterial genome and ask what are all the possible protein coding sequences that exist there. And we can apply some filters. So for example, just even if your genome sequences was typed up by a room full of monkeys and just completely random, that on average the distance between a start codon and a stop codon is predictable. So the random length of a what we call an open reading frame is predictable because there are 64 codons and there are 3 stop codons. And so approximately once every codon, once you find an AUG start codon, you're going to run into a stop codon just at random. And so, however, if it goes for many, many, many codons beyond that, then you have to think that biologically there is something significant here about this coding sequence. So even just ab initio, without any other information other than understanding the genetic code, we can make predictions about lots of protein coding genes, for example. And then, of course, we can ask, well, does the protein with that amino acid sequence or something like that exist in any other organism? Similarly, there are other kinds of genes, of course, and those other kinds of genes, 2 of them that are very interesting, for example, and very important, like the ribosomal RNA genes or the tRNA genes. These things have two things in common. One is, particularly for the ribosomal RNA genes, they are highly conserved in sequence. Particularly, there are stretches of those sequences that are similar across, for example, all bacteria or all archaea or all eukaryotes. And so it's very easy to identify regions of the genome that encode these ribosomal RNA sequences. And the second thing about ribosomal RNA sequences, and is also true about tRNA molecules, the genes for those, is that their RNA folds into a particular pattern based again on complementary base pairing principles. So C's pairing with G's and A's pairing with U's that predict that and produce a reproducible secondary structure. Again, you can imagine that a computer algorithm can search through vast sequences for those kinds of structures. And so you will, in your analysis of your genomes, and some of the annotation software that you will run, will be able to look through, for example, one of the components of your PROCA algorithm will look for all the possible tRNA molecules. And it uses the fact that this tRNA molecules are highly conserved in their ability to form this secondary structure. Okay, it's very predictable, particularly bacterial tRNAs and eukaryotic tRNAs are very conserved in their structure. And not only can it find very accurately these tRNAs, but because of their structure, it can look at the anticodon and tell you what tRNA they are. So it can tell you that it's a tRNA for leucine or a tRNA for serine, and can annotate those. And so that's the point here is that there are aspects and things that we know about cell biology and about genomes and genome structure that allow us to make very strong predictions about the functional capacity and the functional aspects of a genome. Okay. The final thing I want to do before I close with you and you begin your process of analyzing your data is I want to convey to you that what you're going to learn in terms of the tools of bioinformatics and the way that you're going to approach your data is really as it is done in practice out in the real world, essentially. So most tools for bioinformatics are open source, and which means that they are tools that are free, and they're available for you to either download and use, or in the context of you running things on our server, for us to put on our server and run. And so all of the software that you will be using for your analysis are in fact open source, published open source software. Right? The other thing is that understanding how these tools work, we will talk to you about it, and I've given you a little brief sort of high-level overview of things, but there's a tremendous amount of information for how these tools work on the web, and you should use those resources. The other thing is that, as you will notice, and it may be obvious to you already, is that most of these tools are run from what you might call a command line or a bash environment. So this is a very terse environment for some people when they're operating with their computers. But that is the way bioinformatics is done, and there's a whole bunch of reasons for that. But we understand that operating in the command line environment, while it's not hard, it is foreign, and it represents a hurdle. And so one of the first things that you'll be doing as part of this process is understanding that bash environment and how to work in that environment. And so I look forward to seeing your genomes assembled. Good luck.